আসসালামু আলাইকুম ওয়া রাহমাতুল্লাহ। আমরা ইমিউনেট ইমিউনিটির উপরে আগে একটা লেকচার নিয়েছিলাম একটু আগে, এখন আরেকটা লেকচার নেব, সেটাই হলো ইনেট ইমিউনিটি রোল অফ রিকগনিশন মলিকুল। Innate immunity, role of recognition molecules and damaged self by the innate immune system. Review questions after this lecture. We will have to face these questions. What is immunity? What are the outcomes of immunity? And lastly, what are the role of importance of pattern recognition receptors and pathogen-associated recognition, pathogen-associated pattern. Let us start with, how does the host organism detect the presence of infectious agent and dispose of them without destroying cell tissues? How non-specific is innate immunity really? Innate immunity has long been thought as a non-specific immune response, characterized by engulfment and digestion of microorganisms by macrophages and leukocytes, just non-specifically. Now, seeing that innate immunity has also considerable specificity, of course, much less than the innate acquired immunity. It can also recognize self and non-self, and it can discriminate between pathogens broadly, not individual pathogens. Innate immune response also shown to be a prerequisite for triggering acquired or innate acquired or adaptive immunity. There are receptors for innate immune system recognizes microbes and damaged cells by pattern recognition receptors. These pattern recognition receptors are located in cell surface, inside the cells, and in blood and body secretions also. Innate immune system do not react against normal healthy cells and tissues. So, innate immune system recognizes microbes and damaged cells by pattern recognition receptor. You will have to remember this. And these pattern recognition receptors are located in the host cell, that is cell surface, inside the cells, and there are some soluble pattern recognition receptors also. And innate immune system do not react against normal healthy cells or tissues. So, molecular pattern we can describe. There are two types of molecular pattern. One is pathogen-associated molecular pattern. These structures are produced by organism, but not host cell, and they are recognized by and stimulated by the innate immune system. Innate immune system has pattern recognition receptor. For example, bacteria has got lipopolysaccharide, has got flagella, capsule. These structures are not present in our cell, our human beings. Similarly, bacteria has viral double-stranded DNA. We don't have that. Similar, many structures. There are another types of pattern recognition receptor that are damage-associated molecular pattern, DAMP, PMP, DAMP. So, passive damage-associated molecular patterns mean our cells and tissues become old, may be damaged by wear and tear. They are recognized by the innate immune system. These endogenous molecules that are produced by or released from damaged cell or dying cell bind to the pattern recognition receptor and stimulate innate immune response. For example, stress-induced protein, nuclear protein, etc. Innate immune recognition via pattern. So, all multicellular organisms are able to recognize and eliminate pathogen. And despite their extreme heterogeneity, pathogens share highly conserved molecules. These are called pathogen-associated molecular pattern. I uttered this term many times just to imprint in your mind. And host cells do not share pathogen-associated molecular pattern. Host cell do not have peptidoglycan. Host cell do not have lipopolysaccharide, do not have flagella, all these things. And these pathogen-associated, because they are present in the pathogen, molecular pattern are recognized by pattern recognition receptor. Pattern recognition. So main keyword is here molecular pattern. So there is one challenge. How can the host discriminate large numbers of diverse pathogens from each other or from self using a restricted number of receptors? Solution is these unique structures like peptidoglycan that is uniquely present only in bacteria. So our immune system can recognize it, and you can say this is a, by recognizing the peptidoglycan, it can say it is a bacteria. By recognizing double-stranded RNA, yes, it is a bacteria, like this. So what are the examples of pathogen-associated molecular patterns? These are lipopolysaccharide from the gram-negative cell wall, peptidoglycan, both gram-positive and gram-negative cell wall, lipotecoic acid found mainly in gram-positive cell wall, mannose-rich glycans, they are present in microbial glycoproteins, but rare in humans. Flagellin found in bacterial flagella, bacterial nucleic acid. Double-stranded RNA unique to most viruses. Lipotecoic acid, glycolipids, zymosan present in the yeast cell. Phosphoryl choline, other lipids common to microbial membranes, but not to host membrane. Let us see the example of pattern recognition receptors. Pattern recognition receptors are molecules or receptors that recognize this pathogen-associated molecular pattern. And they are soluble, secreted proteins such as phycolin, C-reactive protein, may be on the, they may present on the cell surface and sometimes inside the cell also. There are functionally two classes of cell surface pattern recognition receptors, cell surface recognition. One is, say, endocytic pattern recognition receptors. The very name suggests combining with this receptor will lead to endocytosis. There are mannose receptor and scavenger receptors. There are signaling pattern recognition receptors, and their name suggests that binding with this receptor will lead to give some signals to the nucleus, and these receptors, examples are toll-like receptor, CD14 receptor. Let us examine the endocytic pattern recognition receptors. Endocytic pattern recognition receptors are found on the surface of the phagocytic cells, and they promote attachment of the microorganism to phagocytes. On the surface of microorganism, there are pathogen-associated molecular pattern, and on the surface of the phagocytic cell, this endocytic pattern recognition receptor. So when pathogen-associated molecular pattern will bind with the endocytic pattern recognition receptor, there will be engulfment and destruction. Previously thought that it happens automatically. Now we know that it is a very organized process. There are mannose receptor, scavenger receptors, and opsonin receptors. Endocytic, among the mannose receptors bind to mannose and phacose group on microbial glycoprotein and glycolipid. Scavenger receptors, they bind to the lipopolysaccharide, peptidoglycan, and teichoic acid, but the result is same. Now, opsonin receptors, the name suggests that binding with these receptors will lead to opsonization. There are acute fast proteins, complement pathway, surfactant proteins. In this thing, these are the examples where opsonization occurs. You see, opsonins are soluble molecules produced as a part of body's immune defense that binds the microbes to the phagocytes. One portion of the binds to the pathogen-associated molecular pattern on the microbial surface. Another portion binds with the specific receptor, that is pattern recognition receptors, which in this case is opsonin receptors. For example, these acute fast proteins, you know, classical example is C-reactive protein. In acute infections, C-reactive protein increases. What it does, it binds to the phosphoryl choline on the bacterial membrane and phosphatidyl methanolamine on fungal membrane. And on the other side, it binds to the opsonin receptors present in the phagocytic cells, and thereby there is opsonization, that is facilitated phagocytosis. In the complement pathway, during the activation, there are C3b and C4b. Their action is also same. And surfactant proteins, we have discussed, this is also a component of innate immunity present in the lung, and their function is also similar to C-reactive protein, C3b, C4b, and similar opsonization. Now, a little bit about signaling pattern recognition receptors. Signaling pattern recognition receptors lead to the synthesis and secretion of intracellular regulatory molecules, such as cytokines. Cytokines we will describe in details in a separate lecture. This cytokine play a crucial role in initiating innate immunity and acquired immunity. Basically, it is a link between innate immunity and acquired immunity. There are two types of signaling pattern recognition receptor, toll-like receptor and CD14. Toll-like receptor has got a story we will tell in other time. And there were some animation. These are some animation. If it works, you will find very interesting. You see a bacteria by its pathogen-associated molecular pattern binds with the endocytic pattern recognition receptors, resulting in phagocytosis. So you understand, again, you can see this. Pathogen-associated molecular pattern present in the bacterial cell binds out, and there's endocytosis. Next one, toll-like receptors. These are transmembrane proteins. Transmembrane means one side outside the membrane, another is inside the membrane. And they binding of microbial molecule to these toll-like receptors transmits a signal to the cell's nucleus, including expression of genes coding for the synthesis of intracellular regulatory molecules such as cytokine. These cytokines in turn bind to the cytokine receptors of the defense cell that trigger innate immune defense as well as such as inflammation, fever, phagocytosis, and provide immediate response, and also links with the acquired immunity. There are about 10 toll-like receptors, 1 to 10. They expressed differentially in immune cells, low level, also expressed on other cell types, for example, endothelial cells, respond to different stimuli. Different means both immunological and inflammatory, and they are inducible. And here you see the examples of toll-like receptors, toll-like receptor for lipopolysaccharide, toll-like receptor for double-stranded RNA, toll-like receptor for flagellin, peptidoglycan, so many things. And which toll-like receptor will bind with which structure? This is given in this table, you will just see. This is the animation, how toll-like receptors act. You see again, pathogen-associated molecular pattern. It binds with the signaling receptor, and it leads to formation of cytokine. These cytokines will induce, you see, previously there was endocytosis, but here it will not lead to endocytosis. Just it will lead to activation of cytokine gene. This cytokine will produce receptor. This how toll-like receptor links innate immunity to acquired immunity. You see, when a pathogen by its pathogen-associated molecular pattern binds with toll-like receptor, which is signaling receptor. Signaling receptor, it will lead to production of interleukin. But if it binds with the endocytic pattern receptor, that will lead to phagocytosis. This interleukin and the processed antigen, when it will be present to the naive T cell, T helper cell, along with the MHC 1 or 2, we will take separate class on MHC. And if it gives co-stimulatory signal like interleukin 1, there will be activation of helper T cell. This helper T cell will again produce cytokine 2, which will act on itself. Finally, there may be T helper, if it is endogenous antigen, T helper 1, that leads to interferon gamma and cell-mate immunity. And if it is T helper 2, that will go to the antibody-mediated immunity. So summary, there are scavenger receptors, there are toll-like receptors, and these play a very important role in innate immunity as well as linking of innate immunity to acquired immunity. There are some differences. It is also discussed in the previous lecture, innate immunity and adaptive immunity. Pathogen, in the innate immunity, pathogens are recognized by pattern recognition receptors. But in case of acquired immunity, it is recognized by B cell receptor present on the B cell surface or T cell receptors present on the T cell surface. Receptors have a broad specificity. They recognize broadly, say, peptidoglycan of all bacteria or flagellin of all motile bacteria, like this. So pattern recognition receptor recognizes pathogen-associated molecular pattern. But receptor in the B cell or T cell are highly specific. They recognize the specific epitope after processing, and in B cell without processing. Innate immunity gives rise to immediate response, but adaptive immunity slow response because of the need for the clones of the responding to develop. There is no memory in the innate immunity, but memory is usually present in the adaptive immunity. So best wishes for you, and we will discuss the question answer in a final session when just we will answer your questions regarding these lectures. Thank you very much.